My name is Ariel first, and I'm finishing my third year as an A.O. Beckman postdoctoral fellow at the University of California, Berkeley in the lab of Matt Francis. Arnold Beckman was an inventor and innovator who combined electrochemistry and more fundamental chemistry to bring to kind of the general population scientific instrumentations. Arnold Beckman and his wife Mabel founded the Arnold and Mabel Beckman Foundation, which is committed to philanthropy and pushing forward scientific frontiers. Winning the A.O. Beckman postdoctoral fellowship has really enabled me to be an independent scientist much earlier than I would have otherwise. Basically, the funding through the Beckman Foundation has allowed me complete freedom in my postdoctoral research and has allowed me to pursue questions that really interest me. And so it's really enabled me to have a collaborative postdoc with Matt Francis. So because of the freedom the A.O. Beckman postdoctoral fellowship has given me, I have had the opportunity to both attend many scientific conferences, which has allowed me to grow my network and also has enabled me to train both undergraduate and graduate scientists. So through this fellowship, I've trained 2 undergraduates and also 2 graduate students who will both be continuing projects that I will leave behind when I start my independent career. So my research has always focused on combining electrochemistry with biology to improve sensors. So we've focused on sensors in my graduate work mostly for diagnostic devices. In my postdoc, I've worked on developing new platforms for clean energy as well as environmental sensors. So one of the reasons that I've focused on electrochemistry throughout my research is because it's an exceptionally powerful tool. Electrochemistry is really the only technique that allows you to simultaneously monitor sensitively and specifically interactions as well as catalyze chemical transformations using a single platform. So most people are familiar with biofuels as an alternative to kind of fossil fuels for clean energy. However, even those biofuels have their drawbacks. Many of them are derived from crops, so this causes us to rely on land that was once used to farm food to now make biofuels. It also takes up precious water resources and can lead to things like deforestation and excessive use of chemicals. So one alternative to these biofuels is actually using microbes to create energy. And these microbes can actually use wastewater to create energy, effectively generating electricity from waste. So my first project sought to combat these key problems with microbial fuel cells by using the power of DNA. So the first main issue with these microbial fuel cells is the actual growth and attachment of the cells to the electrode surface. And so by using DNA hybridization, we've been able to improve how those cells interact with the surface. So what we do is we label the electrode surface with one strand of DNA, and these microbes with the complementary strand. And then through DNA hybridization, which is an intrinsic property of DNA, these cells will actually stick to the surface, generating layers of cells very quickly, much more quickly than if they were grown naturally. And so we've been able to use this DNA that's already attaching the cells to the surface to actually be a conduit for electricity between the cells and the electrode, improving current generation and making it significantly more consistent. So overall, what we've been able to accomplish is the generation of these microbial modified electrodes that are much more consistent, and we can actually use them much more rapidly. So what this generally would allow us to do is the more broad implementation of microbial fuel cells, especially in things like wastewater treatment plants, to actually use that wastewater to generate electricity. So the second focus of my postdoctoral research was to develop an environmental sensor to monitor for these pollutants called xenoestrogens. So xenoestrogens are a class of compound that's part of a family called endocrine disruptors. And these compounds actually can enter your body and act like hormones. And so they're very problematic because they can generate kind of off-target biological effects. And these are thought to include things like obesity and diabetes and even some cancers. So we sought to target a family of these that can bind to your estrogen receptor. So the proteins in your body that normally bind estrogen also bind these pollutants. And these pollutants include many different man-made chemicals and some drugs, including chemotherapeutics. Other compounds in this family include things like BPA or bisphenol A, and many people are familiar with this because it's used extensively in kitchenware, in things like Tupperware containers, as well as the lining of aluminum cans. And it's even known to leach into food at low concentrations from those cans. So what we sought to do was to create a very rapid, disposable, portable sensor to detect these compounds in complex solutions in environmental situations. And the way we did this was by combining the best aspects of current methods of detection. So we combined the rapid readout of biosensors with the kind of generality of cell-based assays in order to create an electrochemical sandwich assay. It's an E. coli that actually has that human estrogen receptor expressed on its surface. And so when a small molecule pollutant like a xenoestrogen, like BPA, binds to our E. coli protein, that whole E. coli binds to our surface and we can detect that using impedance electrochemistry. And so overall, what we've been able to do with the sensor is to develop a platform that gives you a readout that's a measure of the total biological activity, even in these complex solutions, so that it allows you to measure things such as the presence of these compounds in things in your kitchen, or you could even think about environmental pollutants such as things that are found in groundwater and in rivers and streams that could affect not only humans but also animal populations. So one of the key advantages of this sensor platform is its low cost and portability. It's also disposable. So currently, the electrode, the protein that modifies the electrode, and the E. coli all cost us less than $1 to produce. So we envision at this cost point, this platform being effective for both things like governmental regulation, as well as kind of industrial plastics companies, as well as for individual use. You could envision this kit being sold on a shelf at Walgreens where people could go and then test their own kitchen products to ensure that they're not getting chemicals leaching into their food. So the A.O. Beckman Postdoctoral Fellowship has really allowed me to do these projects. Without this funding, I would not have been able to pursue them. And these projects have both pushed the frontiers of electrochemistry forward and electrochemical sensors forward, and they've also provided very fertile training ground for several undergrads as well as graduate students while bringing a new skill set into the Francis Group that will stay even after I leave. I want to thank the Beckman Foundation for providing me with this funding that has really given me unparalleled training as a postdoc.